Hi, everyone, and welcome to episode 841 of People Behind the Science. Today, we are excited to reshare our episode with our guest, Dr. Ian Winship. Listeners, Ian is interested in understanding how we can reduce the damage early after stroke and ways we can improve recovery in people who had a stroke a long time ago. His research also examines changes in the brain that lead to symptoms in other brain disorders like schizophrenia. And in this interview, Ian gives us an inside look at his life and research. And I hope you enjoy this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of the People Behind the Science podcast. Today, I am looking forward to introducing you all to our guest, Dr. Ian Winship. So, Ian, welcome to the show today. How are you?
I'm doing very well. How are you doing?
I'm doing great, and I'm excited to chat with you about your life and your work. But before we dive into the details, Ian, I do want to spend some time here up front to tell our listeners a little bit about who you are and what you do. So listeners, Ian is an associate professor and the former Alberta Innovates Health Solutions Scholar in the Department of Psychiatry at the University of Alberta. He is also director of the Neurochemical Research Unit there. Ian received his bachelor's and doctoral degrees in psychology from the University of Alberta, and afterwards he conducted postdoctoral research at the University of British Columbia before returning to the University of Alberta to join the faculty there. And in our interview today, Ian is going to talk a little bit more about his life and his science. So we're excited to learn about both sides of you. And I'd love to start by talking about life outside of science. So how do you spend, I'm sure, your copious amounts of free time, Ian?
Free time seems to be a foreign concept sometimes.
Mm-hmm.
Well, I'm Canadian, so very typically, I think pretty much fall through spring, I spend most of my time in ice rinks. I have two kids, a daughter in grade 4 and a son in grade 1. And they both play ice sports. So my daughter plays a sport called ringette, which is fairly popular in Canada, but not too much elsewhere. It's kind of like hockey. It's an ice sport played by girls and women here. And then my son plays hockey, and I coach with both. So that takes a tremendous amount of time. And then I also play on my own recreational hockey team. So currently, it seems like I spend most of my time outside of work in an ice rink somewhere. In the summer, we don't spend our time in ice rinks. So I just like to be outside. We travel. We like to go to the mountains and to the beach when we can.
Excellent. So have you been following the Olympic hockey matches?
I love the Winter Olympics.
Mm-hmm.
We have the Edmonton Oilers here. I'm a big Oilers fan and an NHL fan. So I watch a lot of hockey, but I like Winter Olympics. I like to watch the kind of random sports that I normally don't see. You watch luge and snowboard, slope style, all this kind of stuff. So I haven't watched a hockey game yet. I'm sure I will.
Fantastic. Well, great to get a window into how you spend your time when you're not hard at work. But I would love to chat about your work next, Ian. So can you tell us a little bit about how you describe what you do to someone who's not familiar with your research?
Sure. If there's somebody from outside academia, generally if I say I'm a professor, then their first question is usually, what do you teach?
Mm-hmm.
So then I actually don't teach a ton. I'm in the Department of Psychiatry, so I teach a bit of neuroscience and I teach a bit of psychiatry, but not a ton. So then I usually say, well, actually, I'm mostly a scientist. And I start very general and trying to gauge how interested they actually are. So I'll say I'm a scientist and I study the brain. And then if they follow up questions, I'll say that I'm interested in stroke and how we can develop better ways to reduce the damage early after a stroke, or ways that we can improve recovery for those that had their stroke a long time ago. And then I'm also interested in other brain disorders like schizophrenia and what it is that changes in the brain that leads to some of the symptoms we see. And then if they're really interested, then I'll start to get into the hows, what we do, focusing on some of the cool techniques and things we do to basically assess those things.
Excellent. I like that you're come equipped with these different levels of explanations. So
yeah, I have a lot of experience talking to people who come from an entirely different sphere of the world.
They want the high level view. I appreciate that.
Yeah.
So now, can you tell us, I think as a scientist, there's a lot to get done. There's a lot of things to keep you motivated in terms of the things you're curious about. But are there particular quotes or sayings or phrases or things that really keep you motivated or inspired?
There's a lot of quotes that I really like that I like to keep in the back of my mind. I'm a big fan of Carl Sagan, so there's a lot of things. You can go through his books and find all these inspirational or thought-provoking quotes. In terms of science, one of the ones I like is he has a quote about how we can judge our progress by the courage of our questions and the depth of our answers. and by our willingness to embrace what is true rather than what feels good. And I think that's important in science and important in life. That's important that you accept what's true, that you don't just fit things to your own narrative. Beyond that, I also like the quote by Charles Darwin that ignorance more frequently begets confidence than does knowledge. Not so much for the second part of the quote, which is kind of in reference to those who don't believe in science and the scientific method, but just as a reminder that if you really feel confident about something, you should really look at it again and make sure that you're not missing something, that you're not letting your excitement cloud your view of how much you really know. And then outside of science, again, there's so many amazing quotes. I really like Martin Luther King Jr. and his quote, "Darkness cannot drive out darkness, only light can do that. Hate cannot drive out hate, only love can do that," which these days seems fairly topical.
Absolutely. Well, I think in talking about these quotes, you mentioned so many inspirational figures in and outside of science. Darwin, Sagan, MLK, these people are huge inspirations for so many people. And I love hearing a little bit more about the mentors, the role models, the people who inspire scientists. So do you have any people who really come to mind who maybe inspired you along your career?
I have many mentors, basically everyone I've worked with, particularly the supervisors, but peers as well. I think it's important, not just in science, in life in general, to look at people and acknowledge that everybody has strengths and weaknesses. And as you go through your career and your life, to try and perhaps emulate the strengths and potentially avoid the weaknesses that other people have, since we all have our own. We don't need to borrow anyone else's.
Right.
I think I learned a lot from all my mentors, my supervisors. So I started here with Douglas Wiley in psychology, doing kind of visual neuroscience. And there, I mean, I think what I really took from him is that science should be fun and you should be enthusiastic about your work, enthusiastic about the questions you're asking. Sometimes people get bogged down in the day-to-day chore of actually getting the data, and they should remain excited, I guess, about the research question. From Tim Murphy at UBC during my postdoc, I really learned to not let technology or technical limitations stop you. His lab is really cutting edge in terms of developing imaging techniques and much beyond imaging now. But when I was there, I came from a background where I kind of learned a few skills and I used them every day in my lab. And in Tim's lab, I'll never forget, was one example. One day I was doing a particular kind of imaging that allows us to image calcium transients in neurons. So it's kind of cellular imaging of activity. And we decided we needed a regional imaging tool. So a tool that would allow us to image over a larger area. And we knew about intrinsic optical signal imaging, which is a method that you can do just that. So he just came into the lab with a camera that he'd found in his office, I think, and we basically sat down at one of the microscopes, and with his guidance, we started to put it together and then kind of said, okay, well, you're on your own now, finish it up. Mm-hmm. With help from other people in the lab, we set it up pretty much within a day or two. And I think that was the first time I'd had that kind of experience where I was like, well, we need something new. We're not going to delay. We're just going to do it now. So let's figure out how to go ahead. And then I guess if I was going to pick someone who'd be a role model that I didn't work with, there's a fairly famous neuroscientist at the University of Lethbridge named Brian Kolb. So the University of Lethbridge is a relatively small university, but he is world known. He's a fellow of the Royal Society of Canada and the officer of the Order of Canada. And he built this truly remarkable career at, again, a fairly small university that actually didn't even have a graduate program in neuroscience until relatively recently. But he did it by building an exemplary reputation as a behavioral neuroscientist, an expert in brain plasticity, and he kind of brought attention and collaborators to him effectively. And then the reason I would pick him as a role model is he's built a pretty incredible facility there, and he's recruited a lot of people, and he's somebody who really seems to take pride in the success of his peers and his trainees, and basically has built kind of a legacy of supporting those people.
Excellent. Well, I love a lot of these traits that you pointed out as the sort of traits you would borrow from mentors, from people who've inspired you along your career path. And I'm curious to hear about where this career path began. Is there a particular memory that sticks out in your mind as maybe the first time you started thinking about science or getting excited about it?
Definitely in high school. I guess it would have been grade 10. I really started to get into biology. Of all the courses I took, it was clearly my favorite, to the point where I took my last biology course in grade 11. I sped up the process because I was really enthusiastic about it. Because then I had to deal with all the physics and chemistry, which I wasn't as enthusiastic about.
Right.
But I still took them. Certainly, I shouldn't say that. I mean, I'm enthusiastic about physics and chemistry. I just don't find that it's necessarily a natural talent of mine to think in those ways.
I struggle there too, so it's fine.
So yeah, I really liked biology. I liked science in general. So I didn't have a firm plan. I certainly didn't set out thinking I was going to be a scientist at that point. I think I thought I wanted to be a physiotherapist, but I rolled in general science. Again, really liked biology. got exposed to all the different sciences and lab work. And then I transferred into an honors physiology program, which to this point in my life, I had never really had to work too hard in school. I wasn't getting the very best grades. I was doing pretty well. I was doing it without really good organization or study skills. I went into an honors program that did not work very well in an honors program. So I only spent 1 year in that program. And then I had taken what we call science psychology courses. So here at the U of A, at least currently, the psychology program is split into a program that's delivered mostly by the Faculty of Arts and one by the Faculty of Science. And the science psychology is really about brain and behavior. And I really liked that. So I went into a specialization in that. And then at that point, I met Dr. Wiley, who I'd mentioned earlier. And I did an undergraduate research course with him, so working in his lab. And the funny thing about that is I applied to do this research course, and he initially looked at my transcripts, which again weren't great from the year before. And he said, no, I don't think your grades are high enough to do this. And then his story is he came back the next day and I was just working in the lab. It's not technically true, but I did have to kind of negotiate my way into it and convince him that I'd had a bad course the year before, but I was a pretty smart guy and I was enthusiastic. So yeah, I went into his lab, and I think it was at that point we were doing tract tracing, so injecting neuroanatomical tracers and looking at where different groups of cells project, some basic microscopy. And I just really fell in love with looking at the cells of the brain, their projections, and I was really excited about that. And we did it with a little bit of electrophysiology too, so you could record cells on the way to do your injection. And I really thought it was fascinating. I think at that point is when I decided that it was something I wanted to pursue more. Mm-hmm. So then I did my graduate program with him and continued to kind of explore this visual pathways. And the good news was I published quite a bit. I had some good success with scholarship applications and that kind of thing. So I started to feel like, oh, maybe I can do this. So I decided to do a postdoc. And I'm not normally, I don't think, the best for planning ahead. But in this case, I did think about it. And I think I made a good choice because I decided two things. I'd always done curiosity-driven discovery research in Doug's lab, which I totally value and I think is absolutely essential to progress. But moving on to my postdoc, I thought, okay, well, I want to do some research that has some relation to brain disorders. And the other thing, well, I know that it's hard to get a job, so I wanted to find a technique that was unique, something that would set me apart from other people who were applying for a job. And then the third criteria was I was married by this point and my wife was in Edmonton and had a good job already. So either it had to be a place where she could get a job or a place that wasn't too far away. Mm-hmm. That was a bit more limiting, but I was fortunate because when I looked around for supervisors, I found Tim Murphy's website initially, and then I emailed with him. And he basically had all the things I was looking for. So he is now one of the world leaders in 2-photon microscopy and optogenetics and other types of imaging. And he had started that at that point, but he was also working in stroke models, looking at a number of things after stroke, but particular changes in neurons after stroke. Mm-hmm. So I emailed him, and despite the fact that I didn't really have any direct relevant, he took me on. So I was excited about that. And yeah, I went there and immediately kind of jumped into this foreign world of, I hadn't even worked much with mice and with imaging, and really an environment, as I mentioned, where you were expected to work at things on your own and figure it out as needed. So that was a really great experience. I was very fortunate because I'd collaborated with a number of people here at U of A that I had an advocate who was enthusiastic about bringing me back. So I think I got extremely lucky in that I went into Tim's lab, I started to have some success, and a position became available here that Catherine Todd, who's a professor in the department here, really wanted to try and recruit me back to. So I interviewed here after a pretty short postdoc and was able to get the job and came here in 2007 after just a couple of years of postdocing, which was extremely exciting. Moving back home was not necessarily something that I thought I would be able to do. My friends and family are here and my wife was still living here, so that was ideal. But then it was a challenge because I had such a short postdoc. You kind of learn after you start that the 10-year clock and the expectations start as soon as you start. Mm-hmm. So even if you're somebody like me that is still kind of wrapping up postdoctoral work and getting out papers, it adds a little bit of challenge to your day, I guess. But I'm still here 10 years later.
It's like everything worked out well, it sounds like. So
yeah, I eventually got some grants and I got tenure. So that's good.
So life is good. I like it. You brought up so many fantastic points in this story of your career. This idea of not willing to take no for an answer, even for that first lab experience, showing that persistence and really just getting in there and making it happen, I think is so important because that kind of sets you up for all the experiences that come later. I think getting that early research experience can make all the difference.
Absolutely. And you have to believe in yourself. I think one of the really cool things about science, the general public probably thinks most scientists are just data heads, really smart people. But I think there's a lot of ways you can get into science and be good at it. And I think if you're just a good thinker and you believe in yourself and you know that you have good ideas and you can evaluate things well and critically think about topics, I think that's really all you need to get started. It isn't easy. You have to find the balance between believing in yourself and then also, as I mentioned before, questioning yourself and making sure that you're not overconfident. But I think science is truly a field that can accept just about anybody, but it's not always easy to get into, and it's not easy to get a foothold and kind of build your own niche in.
Absolutely. Well, I think you had good strategies and planning going into it. I love that you had this checkbox of all the things you were looking for in a postdoc, and then your skills and fortune came into play then in finding that perfect faculty position. And now you are back at home, like you said, at the University of Alberta. And I know you're working on a lot of exciting projects, Ian. So is there one you want to tell us a little bit about in detail today?
Most of the work that I've done here is built on what I started in my postdoc. But then, as you do when you move into your own lab, you have to make yourself distinct and unique. So in stroke, I've kind of built a niche looking at blood flow in the brain and tools that we can use or treatments that we can use to basically improve blood flow early after a stroke to reduce damage. And then the other kind of unique part of my stroke research is I'm still interested in plasticity and how the brain rewires itself after stroke, which is what I started with Tim. But now we're looking at a couple different things. We're treating in the spinal cord as opposed to in the brain. And we're actually looking at long-term disability. So we're looking a long time after the stroke. Those are cool things. But then I really like new ideas, new things. So the newest thing in my lab is we started a collaboration with a guy at the University of Saskatchewan named John Howland to look at a model of schizophrenia. And the model we use, I think, is really cool. It's called the maternal immune activation model. So this is basically what you do is you take, in this case, it's rats, you take a pregnant rat and you basically inject her with something that makes her sick for a couple days. But she recovers, has the kids at the normal gestational point, and initially the children look fine. But this model actually builds on epidemiological data that shows if you look at populations of humans where there was a severe epidemic or even a really stressful period like a civil war, and you look at mothers that were at a certain stage of their pregnancy and their offspring, if you look at those offspring or children as adolescents, you see that they have a lot higher incidence of mental illness, including schizophrenia. So this maternal immune activation model is a model of schizophrenia. And so when you look at the offspring of those mothers at late adolescent, early adulthood point for a rat, they have all these behavioral phenotypes which match what we think is the rat model or rat phenotype for schizophrenia.
Interesting.
So I find that model super fascinating. What we're trying to do is determine if there is a core feature or core change in the brain that leads to some of the cognitive impairments. So we're particularly interested in these structures in the extracellular matrix that surrounds all the cells of the brain called perineuronal nets. They kind of wrap around certain subsets of neurons. And we found that in these rats, if you look as they age, they have normal perineural nets in the prefrontal cortex. That's an area of the brain that's important for cognitive function. But when they reach that late adolescent, early adulthood point, then they have a significant deficit relative to controls. And we've since done some experiments looking at, well, if we just manipulate the perineural nets in just that area of the brain, is there a cognitive impairment? And there seems to be. So yeah, we're interested in whether or not that's something that is going to be common across all the different kind of genetic mutations and environmental circumstances that can lead to a person developing schizophrenia. Is that something that's kind of a core feature? So we're going to use the imaging tools we have and some optogenetics and that kind of thing to look at what happens in the brain when you lose these perineural nets.
Now, this is really cool. So how did you, I guess, move from work that was focused quite a bit on stroke to now starting to pursue this idea in schizophrenia?
So my interests, perineural nets are the thing that bring this all together. So perineural nets are made of molecules called chondroitin sulfate proteoglycans, which are core components of the extracellular matrix. Now, in my stroke research, we actually use drugs to degrade these CSPGs because the extracellular matrix inhibits structural plasticity. So in the event of a stroke, you want the brain to be more plastic. You want it to be able to rewire more easily. And these matrix components, CSPGs, will actually act against that. So we use drugs that break them down or manipulate them somehow to remove that inhibition of growth. So after stroke, we think that's a good thing. But we also wondered whether or not perineural nets which are, I guess, a specialized CSPG structure, might be implicated in different brain diseases. And I was watching John Howland. We go to a behavior and brain conference in a small ski town in British Columbia here, which is actually organized by Brian Kolb, the guy I mentioned earlier.
Oh, yeah, yeah.
And he studies this maternal immune activation model, and he studies cognition in rodents. And I just wondered, okay, we know that these structures are important for regulating plasticity in the brain during development and after injury. And there's an idea that the brain becomes kind of aberrantly plastic, perhaps, in different diseases like schizophrenia. So we just thought, well, we should check this out. We looked in the literature, and there was a bit of postmortem data that suggests that, yes, these perineural nets might be changed in different psychiatric disorders, including schizophrenia, but there really wasn't much there. So we thought, well, this is a cool new area. And John is a friend of mine. We wanted to find something to collaborate. So that was about the total justification. We thought, okay, well, we have tissue we can look at, and let's check it out and see. And then I shouldn't say we weren't entirely focused on the prefrontal cortex. That was one of the areas we looked at. I don't want to make it sound like we went looking for one thing and found one thing. We found lots of areas where the perineural nets don't change, but that's the core finding that we have that we've built the rest of our research plan on.
Well, this is a wonderful project. Thanks so much for telling us a little bit more about your work. But I do want to talk a little bit next about some of the challenges and the failures and the struggles you encounter as a scientist. We touched on a few of the early ones in talking about your career, but I think these are so important to share with our listeners who might be going through some of the same things right now. So do you have a favorite failure or struggle that you want to tell us about today, Ian?
I don't know if I have a favorite failure. Personally, I don't think anything works the first time you try it. And when it does, that just means it's not going to work the next 5 times. So that's the hard part. That's why you have to focus on your research questions and why you're doing it, because it always takes, in my experience anyways, and my trainees in my lab here, it takes a lot of time and a lot of perseverance to get to the point where you're effectively and efficiently collecting the data you want. For me, I think probably when I went to Tim's lab at UBC, we're using this 2-photon microscopy, which was new to me. I was working with mice, which were new to me. I was using calcium indicators, which were new to me. And we were also trying to build some new imaging modalities into the systems we had. So I think there were failures at every stage. Imaging wouldn't always work. Surgeries I was not good at the start. We had to design stimuli. We were using a somatosensory stimulus. We had to find stimuli that would work. There were lots of troubles there. And then we had to pick the appropriate amount of stimulation. Then I was getting just huge amounts of data that I did not really know what to do with. So I had to figure out how to analyze all that data. And then on a personal level, I was poor, but trying to commute back and forth between Edmonton every week or a couple weeks, or at least a month to see my wife. So there was a real period there where it was very frustrating. I wouldn't say I was totally happy. And again, I kind of had to remember like why I was doing this, why it was important to me, and why it might be cool if it works out, and really push myself. And then I have to say, for anyone who's moved on to academics or is planning to as a principal investigator, you better realize that failure is going to become the expected outcome in most endeavors. So when you write a grant, when you start a new research project, when you submit a paper, you usually start with the highest impact journal you think you might fit into. You can't go in with the expectation to succeed, or else you're just going to be down all the time. You have to find a way to define your worth as a scientist and as a person that isn't dependent on success every time. You have to remember that just because your paper gets rejected or your grant gets rejected doesn't mean that your ideas aren't good. And then the big thing is you have to learn to filter through all that feedback. The great thing about science is scientists like to talk and they like to write, and you often get lots of feedback on whatever you've done. And you've got to filter through and take the serenity prayer and determine what it is you can change about your ideas that will help address some of those concerns. And then the things you can't do anything about, you got to learn to let go.
Absolutely. Well, I think you mentioned so much helpful advice here in talking about how you work through some of these challenges. And I think you're right. You sort of have to set the expectation that everything is going to fail. And then it's just really exciting if it doesn't that one time.
Yeah. Don't let the failures get down, and then celebrate the successes.
That's right. And speaking of celebrating successes, that's something I really love to do on our program. So Ian, do you have a success story, whether it was a huge win or just a small but very meaningful victory for you, that you want to tell us about today?
Well, I'm still here. I consider that a success.
Congratulations.
Yeah. But actually, I've been here 10 years now, and I think I've reached a stage of my career where I am really starting to get excited when my trainees have success. So most recently, we had a paper that just came out in the Journal of Neuroscience, which is a great journal, and I'm super happy about that on a personal level. But it was also a paper that involved several years of my former PhD student, Anna Wiersma's work. So she spent years This is a behavioral study in rats with rehabilitation. She spent so long working with rats and she has bad allergies. So it was very difficult for her, but she kept pushing, she kept working. So getting it into the journal was exciting. But then we also, this is the first time, largely through her efforts, we've done media. So we've had some interviews with magazines and online articles and some TV features and stuff. And I can't say I'm terribly comfortable with that, but it was really, I think, exciting to see how excited she was about it. And also for her, all of a sudden, her friends and family were seeing that what she does has value. So she was getting that feedback that it's all worth it too. So that's been exciting. I've had good luck the last little while with trainees getting scholarships and all this kind of stuff. So yeah, I think I'm at a point now where I'm really starting to get excited when the people who really spend all the time grinding away and getting these projects started and finished, they're starting to reap the rewards. And that's pretty exciting.
Absolutely. Well, congratulations, Ian, to you and your trainees for all the phenomenal work you've been doing. And I had to chuckle a little bit when you mentioned that you're finally starting to get in the media. I think that's how I found you. So
there you go.
That's wonderful. So can you tell us a little bit about how you celebrate success in your lab?
We're not as good as we should be. So we usually, for a paper or a defense or something that goes well, we'll sit around as a group and we might have some champagne or beer. We try and get together regularly to go out. And usually it's just, even though my lab actually doesn't have a lot of people who drink necessarily, but we try and go to a pub and have some snacks and a drink and talk about it. But my students, I think it's changed a little. I don't know. Maybe it's just my lab. But they all have their own lives. So they like to get together for a while and celebrate. And then I think they want to go celebrate with their close ones or their family and friends. So we have our little lab rituals and we try and make sure it's acknowledged. We go out a bit and I usually have a party or two a year to celebrate, but I wouldn't say anything truly remarkable.
Gotcha. Well, I think it is important just to take that moment to talk about the success, to acknowledge it before just jumping right into the next project. So I'm glad to hear that you do that. And we've talked a little bit about your life outside of science. And one of the things that I'm trying to encourage myself and our listeners to do more of is read for fun. So I love getting book recommendations from all of the guests that we have on our program. So Ian, do you have a book recommendation, whether it's a science book or not, that you want to tell us about today?
I am also trying to read more. My wife is a voracious reader. She's constantly reading and I'm a painfully slow reader. But that said, I do have a lot of favorite books, I guess. I mentioned Carl Sagan. I love Pale Blue Dot, a scientific or kind of scientific book that frames our significance in the universe. Again, I think perspective is important. So I love that book. I also like The God Delusion by Richard Dawkins, and I'm scared to say that because I don't want people who are religious to dislike me. That's not necessarily why I like that book. Richard Dawkins is not a fan of religion, but what I like about it is the idea that any idea is a hypothesis that can be tested. And his example is religion, but it really applies to anything. There's nothing that isn't subject to the weight of evidence. So I think that's an important lesson that especially I think scientists often live that way, but I think just the general public should take that attitude that it's okay to question things. You should question things and you should look for evidence that support or refute your beliefs. And then, as I mentioned with the Carl Sagan quote, if you find evidence that refutes your belief, you've got to accept that as true. Outside of science, I'm a Lord of the Rings nerd. I love those books. I love John Irving books, especially A Prayer for Owen Meany. And then my daughter's in grade 4 and she just read a book called Wonder. Our movie's coming out too. And she also is a voracious reader. She takes after her mom and she loved it. And she really wanted us to read it as well. So I read Wonder. It's written for kids or elementary or junior high kids, but it is a great read and a great book. That's, I think, the most recent or second most recent book I finished, but probably my favorite this year.
Well, for not being a voracious reader, you've given us a lot of great recommendations, Ian. So that's wonderful. And one of the things that we talked about, in addition to some of the opportunities that you've had throughout your career for research, are some of the opportunities you've had to travel, to work with different people, to go to different places for training. And I love talking about travel on our program because I think it's one of the perks that many people don't really think about when you think about a career in science, but you do travel quite a bit to go to conferences and research meetings and work with collaborators. So do you have a favorite place that science has taken you in your career?
I've been lots of places I really like. As you say, most of my friends are from outside of science and they think we live this glamorous existence of going from place to place. I don't travel as much now that with the kids as I used to, but there's a number of places, San Diego, Chicago, New Orleans, Montreal, Quebec City, in North America that I've been. I really enjoyed and I wouldn't have been to otherwise. And the thing I love about travel and science is that many of my friends who are in science have their own labs now. And that's really about the only time we get to see each other. So there's a professional purpose and then there's also the social side, which is amazing. And you can start new collaborations there like John and I did. But probably my favorite place that I've visited would be Munich, Germany. So very unique place. For somebody who was born and raised in Canada, the history of a place like Munich is just hard to comprehend, the age of the city and what it's been through. And I guess the other thing that was unique about that trip was that was one where I went and I didn't know anybody. I was asked to come give a talk relatively early in my career here. So I didn't know anyone. And because of that, I met new people, which was great. But I also did a lot more sightseeing and some of the other stuff that I might not normally do when you're visiting with your acquaintances that you know better. So yeah, I saw a lot of the city and I really loved it there. My son was 6 weeks old. My daughter was 3. So it might also just been that despite the jet lag, I actually got to sleep and that might have also helped.
You were feeling so refreshed. I like it.
Yeah.
So what was your favorite thing to see in Munich?
We went on a tour of the palaces and the opera house. And unfortunately, I can't remember the name of the square, but one of the squares where Hitler had some of his rallies early on prior to the war. And the buildings that were completely destroyed during the war, but then have been restored, I thought was truly remarkable. Again, I'm from this area and we've, knock on wood, never had any major wars. And beyond a tornado, generally speaking, where I live, hasn't had any truly traumatic events. So it's very kind of informative to go to places of the world that were basically destroyed and rebuilt.
Absolutely. And like you said, just the history of the buildings, that was what struck me most, I think, when I went to Europe, is just compared to the United States, you just don't see things that are 5, 6, 7 plus 100 years old. So that's phenomenal. Well, I love that you highlighted too about when you're traveling, the people that you get to travel with, the people that you get to see and sort of have these reunions with at conferences, because I think this human side of science is really important and not something that a lot of people outside of science necessarily appreciate. So I love highlighting this on our program and just talking about some of the funny and fond memories that scientists share together, maybe some of the quirky traditions that spring up in different laboratories. So do you have a story of your own, Ian, along these human side of science lines that you want to tell us about today?
Yeah, I mean, I think scientists are fun people because they are so different. They often have some different personalities, but they're very smart. So I think that makes for a pretty fun group. And again, it's people who tend to spend a lot of time in close quarters and spend a lot of time together. So it forms pretty close bonds and nicknames that can last a lifetime. Mm-hmm. A lot of my favorite times are just sitting around talking science with people. I've mentioned the schizophrenia project. A good friend, Craig Brown at University of Victoria, and then John Howland, who's a friend of mine in Saskatchewan. And we were at this Fernie conference and we just went to, it was a Boston Pizza, I think. We went to talk about this project and whether or not we could put a grant together. And it was one of the funniest conversations because we started out thinking that our ideas were the best and this was a surefire grant that was going to go. Then we talked ourselves completely out of it and decided it was a horrible idea and we didn't have any data and it was never going to work. And then we managed to talk ourselves back into it and went into it with an appropriate level of, yeah, maybe it'll work, maybe it won't.
Give it a try, yeah.
Yeah, but surprisingly, it did work. My favorite part of that story is actually Craig recounting because Craig's not actually involved in the grant, but his recounting of us going through this process in the 7 stages.
The inflation and the deflation. And
Those are the times I really value, just getting together, sitting around, and usually conversations move into and out of science, but we all have kind of a core passion, core interest in the brain, but everybody brings their own perspective. So it's a lot of fun.
Absolutely. Well, wonderful story. And I'm glad you guys did end up sort of talking yourself back up and submitting that project. So
yeah, no, it actually got funded too.
Worked out great. That's wonderful. And we talked about some of the big questions that you're answering, not only with the schizophrenia project, but some of the other work that you've done in the laboratory. And I always love to challenge scientists to sort of think big, think outside the box, because I think so often you feel constrained by limitations in funding or staff or time or technology or feasibility for a project. So if I took away all of the restrictions, Ian, is there one question that you are just dying to know the answer to that you would want to tackle first?
Well, one approach that I guess could be used to answer any of the research questions I'm interested in, but one of the things that struck me is I'm a neuroscientist, I do preclinical research, but I know a lot of clinicians. And one of the issues with some of the approaches that we identify in the lab that look like they're going to be a really effective treatment are not always very practical clinically. There's all these limitations that we don't think about when you actually have somebody and you have a short period of time and limited resources and you don't know if they're going to be in a major city or a rural center or any of that kind of stuff that you don't think about. So a lot of the approaches I've used to treat stroke and even ideas we have about schizophrenia are kind of these focal treatments. You inject something into the brain or the spinal cord at the area you want to treat to improve things. And if I had unlimited resources that included hiring the world's best molecular biologists to come work with me, I would want to work on developing systemic gene therapy. So these are therapies that you could deliver systemically to a person that would then target. So in the case of schizophrenia, you might have it targeting the prefrontal cortex or particular glial cells. and altering their release of perhaps enzymes that degrade this extracellular matrix. And if you had a systemic treatment that was well tolerated and didn't interfere with other areas, then you could give this prophylactically. You give this to young children and they stay on it. In theory, perhaps it prevents the development of the disorder, or at least certain symptoms. And it's the same with stroke research. I think we have these really good ideas about how to harness plasticity at the level of the spinal cord to improve recovery from stroke. But to actually do that would be a fairly invasive procedure in a person. And I think the key would be finding a way to develop a vector that could target just the areas you want and avoid everything else.
Absolutely. So for a lot of these treatments you're looking at, are there side effects that you need to take into consideration because they're not necessarily as targeted as you'd like them to be?
Sure. So our work in this kind of spinal plasticity after stroke, we have to, at this point, inject a compound. The compound that we published was this enzyme chondroitinase ABC, which degrades the matrix. There are others that we're working on as well. Some viral vectors, some are just proteins or enzymes. But you actually have to inject them into the spinal cord. And when you talk to a clinician, they don't necessarily want to do surgery on someone's spine when they've already had a stroke. You don't want to end up making them worse.
Right.
So that's a real limitation. And we have this idea that there might be ways that we can prevent the breakdown of these extracellular matrix structures that we think are relevant to schizophrenia. But that would require a person, a child in this case, taking a drug for many, many years, perhaps their whole life. We don't know what the process is, but if they're going to prevent it, we have ideas on how you might be able to do that, but it's a drug they'd have to take forever. So if it's something you're going to take forever, you don't want it to have really severe side effects. That's one of the problems with a lot of psychiatric medications is the side effects aren't always perceived as worse than the benefits. Mm-hmm. So yeah, I mean, if you're going to have any kind of long-term treatment, you want something that's in theory targeted just to what you want to treat.
Well, you've got your work cut out for you here, Ian. Lots of questions to answer.
Yeah.
Well, you've given me and our listeners a lot to think about today, Ian. And I'd like you to give them one more thing, though, before the day is done, and that is a piece of advice. Is there something that someone told you at some point in your career that really helped you that you can pass on to our listeners today?
In science, I would say publish. I know that people have this publish or perish idea, and it can take the fun out of science if that's what you're thinking about, and if you're paying too much attention to where things get published and what its impact factor is and how many citations it gets. But if you want to move on to the next step, you have to show consistent productivity, and that's every step along the way. And so it's important to remember that projects should have a start and a finish. And at that finish, if you found something, you should publish it. And if it's not publishable, you should move on. Side projects are good. Getting broader ideas about a topic are good, but ultimately you need to show productivity, which is important. In life, my family, my dad in particular, have always emphasized to me that how you treat people is what they'll remember. They're not going to remember your accomplishments in science. They may not remember your big paper or that you got a promotion, but they will remember how you treated them. I think that's important to remember. And that's something I try to enforce for my kids, that when you look back on your life, you're not going to focus so much on what you achieved at work or professionally, but you're going to remember the times you spent with people, and those people will remember you by how you treated them. And actually, there's a quote in Wonder, they do precepts, I think they call them, which are just little
inset quotes.
Yeah. And one of the first ones I think in the book is, when given the choice between being right or being kind, choose kind. I think it's Wayne Dyer that said it. Mm-hmm. In science, that's hard because we always want to be right, and we're always have our ideas and we're trying to prove that ideas are correct. And often there's evidence that might argue against them, and other people have different opinions. But I don't think they're mutually exclusive. You can try and prove yourself right, but you should never forget that how you treat people is ultimately what people will remember.
Absolutely. And I think this last one can apply for both life and science, like you said. Great advice for our listeners. Ian, is there any last piece of advice or a last note of inspiration you want to leave them with today?
I think science is amazing, but I would really caution people not to let it remove them from the rest of their life. You have to work hard. You have to be passionate about what you do. Hopefully you discover amazing things, but there is a world and people outside the lab. On Twitter, there's interesting discussions about how much people work and what you need to do to be successful. Don't believe anybody who tells you need to live in the lab and spend 80 hours working exclusively and lose out on the rest of your life. Even if that were true, it would suggest to me that it's probably not worth it. Love your science, but don't forget there's a world outside the lab.
Well, excellent note to end on today, Ian. If our listeners want to learn more about you and the fantastic work you're doing, where should they go?
I have a website which tells you a bit about my lab. I do have a Twitter account. I think it's at iwin76, but mostly if you go to that, you'll see me retweeting These days, it seems to be mostly things about government funding for science here in Canada and comments on spoilers. So probably not of broad interest necessarily, but generally speaking, email is the best way. You can find my email on my website, and I'm usually happy to chat.
Perfect. Well, listeners, definitely check out Ian's website and get in touch if you have any specific questions. And Ian, thank you so much for joining us on the program and sharing some of your stories about life and science.
Thanks very much. It was a lot of fun.
It was a pleasure to chat with you and great to have you here as well, listeners. We'll see you next time on another episode of People Behind the Science.